All right, really quick. So my name is Lauren Patumtong, and I am the site manager here at the Estuary Center and for the Otter Point Creek component of the Chesapeake Bay National Estuarine Research Reserve. And every summer, with the help of our amazing friends group, the Otter Point Creek Alliance, we are able to host a research intern who can focus on a topic of interest in Otter Point Creek. This program is fantastic because it gives the intern a real world experience, and it allows us to gain knowledge and information on what's happening in our park and our waters. So now I would like to introduce our intern from this past summer, Tim Smoot. Tim is currently a junior at University of Delaware studying environmental science and minoring in marine science. He came to focus on the snakehead research topic, knowing that he had an interest in marine science and animals. And through this experience, he has found a real passion for fisheries science. He has an aptitude for lab work, even when it involves poking fish guts. He has a great understanding of the scientific process, a strong ability to work independently and to create collaborative partnerships, and impressive paddling skills, according to Beck. We're thankful to have Tim as our intern from this past summer, and we're grateful that he focused on this northern snakehead, a species many of us have been curious about since it started showing up in Otter Point Creek. So with that, Tim, I'm going to let you take it from here and show us what you've discovered.
All right. So yeah, so hey everybody. Like Lauren said, my name's Tim, and this past summer I've focused on snakeheads in Otter Point Creek. And so really just what I wanted to do today is share with you what I found while I was out there looking at them, figuring out what I could figure out, poking them in the lab, and hoping to get some answers to some questions that I think we all have. Right, so just before we get started, I thought I would let you guys know what we're going to focus on throughout the presentation. So I'll start off with some information about snakeheads and Otter Point Creek as the study site. So if you don't really know what a snakehead is, don't really know Otter Point Creek, don't worry about it. We'll go over that, get you all familiarized. Secondly, I'll talk about why snakeheads are an important species to study. Like why am I picking them? Why is it something that we need to know about? And specifically, like the questions that I had that I wanted to know going into the summer. I'll talk to you about the different methods I used to figure out, to collect this data about snakeheads, to catch them, to look at them, and really answers those questions that I had. I'll move on to what we learned. I focused on 5 different things, that being the best way to catch them, the best way to collect data on them, what they're eating, how old they are, where they're at in Otter Point Creek, and what they're doing in terms of reproducing. And then I'll wrap it up with some conclusions and directions for future work. So here's a little bit of the introduction about the species that I looked at, and it is the northern snakehead. I will say that there are multiple species of snakeheads, but those are native to Asia, and this species here is the only one that we have in the Chesapeake Bay. So when I refer to them, I'm just going to say snakeheads, but the species I'm talking about is the northern snakehead. And they're a predatory freshwater fish that we have seen here showing up in some of our tributaries. You can kind of see why they're called snakeheads, given the coloration and the shape of their head. You know, this is a fish, but it certainly does look like the head of a snake. One thing that is really interesting about their biology is that they're obligate air breathers. And so basically that means that they have to surface for air. So when they're usually found in these very shallow creeks, smaller waterways, because they can just come up and surface for air. And in fact, if they don't surface for air, then they're in trouble. Generally speaking, they're solitary except during the spawning season, which is May to mid-September. And there's some variation in that because there's some different variation among the different pairs and individuals. When they do spawn, they don't exactly like synchronize their spawning. So you'll have some that start earlier in the season, you know, May, June, and some that maybe don't show up spawning until late in the season. So because of that, you know, you get the variation from May to mid-September, and there is some evidence that they can spawn multiple times per season. I will say it does look like it's relatively limited in how often they're spawning. They can't spawn like every single day because they put a lot of resources and energy into creating a nest and caring for the fry, so they can't really do this all the time. But there is some evidence that they do perhaps spawn maybe 2 or 3 times a summer. Some maybe only once, but some can do that. And when they do spawn, that is when they come together, and then when they're not solitary. So you'll have two adults, a male and a female, that come together and they will pair up. And then what they do is they build a nest. And I'll have some figures of the nest later and some pictures of nests that I found to give you a better idea. But basically what they do is they build this nest and they go through and they clip off some of the vegetation under the water and that floats up to the surface. And then they lay the eggs in that nest. They protect that nest for a few weeks until the eggs hatch. And then even once the eggs hatch, you have these little tiny snakehead fry that travel around in this group as a fry ball. And the two parents, the male and the female adult, both of them will protect that fry ball for a few weeks. So really this whole spawning event takes a lot of resources and a lot of energy. And it just occurs all throughout the summer. So if you're out there in the summer, sometimes you'll see these fry balls, sometimes see the nest, or sometimes see like some baby little fish with a snakehead around them, their snakehead fry being protected by the parents. One thing that I wanted to talk about is that like something that you might see in the news or you see in an article is everybody's like, oh no, like these frankenfish get out and walk on land. And we're concerned about them switching and jumping from creek to creek. And while it is true that these fish can live on land for a little bit of time because they can breathe air, as long as they stay moist, they can survive on land. Really, they don't voluntarily leave the water unless the water conditions are really, really bad. There's this study here that exposed them to a lot of different conditions and gave them this little ramp to see if they would crawl out of the water. And the only time they voluntarily left the water is if it was super acidic, super salty, high concentrations of dissolved carbon dioxide, stuff like that. So it's really not a voluntary thing that they do often, and we wouldn't really expect. Most of the times, as you see a snakehead on land, it's probably because a person left it there or some kind of predator got it out of the water and dropped it on land. Usually they're not voluntarily just crawling out of the water and going wherever. So as I said, snakeheads are native to Eastern Asia, and that's the same for this species. They're not supposed to be here, and they're not from here. And they've really only shown up relatively recently. The first time they were found in Maryland was in a pond in 2002 in Crofton, Maryland. And there were a few adults that were found in there and some fry. And once they were discovered, the pond was promptly poisoned and they were kind of knocked out. But 2 years later, they were found in the Potomac River, which is one of the big tributaries that flows into the Chesapeake Bay. And once they were in there, you can't really control them as easily as you could an isolated pond. So since then, they have spread all throughout the rest of Chesapeake Bay, up into the other rivers, and really made their way just
Snakehead presentation going right now.
So yes, they've kind of made their way all throughout there. And really the mode of introduction is unclear. So there are some people that think maybe they came over from like a live fish market and were released by fishermen to create like a resource here. There's some people that kept them as pets, and maybe it was someone who didn't want to care for their pet anymore and put it out there. There's a religious cultural practice of releasing fish. So maybe it came from there. And really, the honest answer is we don't know how they got here. And some genetic evidence suggests that they were introduced multiple times. So it could have been multiple different of those reasons, but we really don't know how they got here. But we do know that they are here and they have been here since 2004 and they've spread throughout the Chesapeake now. A little bit of background on the study site itself. So this is Otter Point Creek. For those of you familiar with the Estuary Center, that is right here. And then here's Route 40. And then the study site I have drawn out here in white, just to give you an idea of where I was looking for snakeheads at. So that right here, the river flows down this way, flows out here into a much wider area, goes out this way, and then down this way is the Bush River, and then that flows out into the upper Chesapeake Bay. And this is, at this point up in the Chesapeake Bay, it's basically freshwater at this point, but it is still tidally influenced. So you do get the tides, but it is still mostly freshwater. And the upstream portion up here is a lot of these little narrow channels and creeks, plus two little lagoons here. And then you also see in some like the shallower areas along this side, you'll get a lot of really dense vegetation that grows along the bottom. And that's a little bit different than downstream here, which is a much more open area. It is still relatively shallow, despite it being open like that. During a spring low tide, which is the lowest low tide of the month, I've seen this area exposed to the air. So it is still relatively shallow. And same thing during the summer, you get some pretty dense vegetation that grows along the bottom here. And as Lauren pointed out, this is one of the sites within the Chesapeake Bay National Estuarine Research Reserve. And so that's just an area, they're all over the country, these national reserves. And so a lot of research goes on here, and it's managed by Maryland DNR and NOAA, and it's a great place for some research to get done besides the snakehead stuff that I looked at. One of the things that we need to ask before we start getting this is, why do we study snakeheads? So again, I mentioned that they're not native to here, and we don't really know how they're going to impact fish once they're introduced. Whenever you have something that hasn't been here for a while and you bring it into a new habitat, there's a potential that it's going to influence the native fish that are here and all kinds of different processes that go on. And so we really want to understand as much as we can about these fish, how old they are, what are they eating, how big are they getting, where are they living, to figure out, are they going to impact any of these native fish that we really care about, that we need, any resources, commercially important species, things like that. And specifically with this study site, you can see here's a lot of the prior research that is focused on snakeheads, and for good reason. It's focused on the Potomac River a lot and these tributaries down here, because that's where they initially found. But there's not really a whole lot going on up here. And so this is our study site, Otter Point Creek. And it's providing a little bit of a unique perspective to see, are we seeing similar things in the snakeheads up here versus the snakeheads down in the Potomac? Specifically, I mentioned there were 5 things that I wanted to look at, and that is, what's the best way to collect snakehead data in Otter Point Creek? What are they eating? How old are they? Where in Otter Point Creek are they? And is there evidence of a reproducing population? So now we're going to get into how exactly I caught them. And the way that I did that was using an electrofishing boat. So it's this really cool boat that has these anodes that stick out on the front of it, and it sends an electric current into the water. And what that does is it'll hopefully stun the fish. Some of the smaller fish aren't really stunned that well. They just kind of shoot all over. Some of the bigger fish are stunned pretty good, where they stop moving for a little bit. And so what you can do is you just drive the boat along, the fish are stunned, and you can scoop out the snakeheads, leave the rest of the fish be, and after a few seconds, the other fish swim away unharmed. This red path here is where we did the electrofishing. So you can see you came up here and we went in all these channels into the lagoons. We were limited in where we could go because over here during low tide, it's super shallow. So the boat just couldn't get back there. And then coming in this way, there was a down tree, so we couldn't get past that point. But we went on this path 4 different times throughout the summer. And over the course of 4 trips, we caught 23 snakeheads. And so it's really nice when you have the fish caught, because that means that you can get a length, you can get a weight, you can open it up and look at its stomach or different other internal things. And so that's the advantage of this electrofishing survey, is that you get your hands on the fish and you can get a lot more data. So here now is a video just to give you an idea of what electrofishing looks like. So as I said, here are the anodes sticking out, sending a current into the water. You'll see the snakehead pop up here, kind of float across. This is one of the ones that was stunned pretty good. But there it is, pops up, grab it. This one was a little bit big, so I needed an assist on the back end, and got ourselves a snakehead. So fairly straightforward, but not all of them are that straightforward. Sometimes they're moving around, you can't really catch them. Our catch rate was about 50%. So I said we caught 23 snakeheads. That's only about half the ones that we saw electrofishing. Some of them, like I said, they just don't stop moving and they get away before you can get them. Some of them get stuck under logs or just simply out of reach. The nets are only so long. But that's about what we got, and that's pretty much how electrofishing works. In addition to electrofishing, I would go out in either a kayak or on a stand-up paddleboard at least once a week. Usually I went multiple times a week. And I would just go out all through those channels and all throughout Otter Point Creek and just look for snakeheads. And the advantage of this is that I could get into areas where the electrofishing boat couldn't go. So I said it was too shallow in the one spot. There was a down tree. I could navigate around that in my kayak. And so really I could just go out there, sneak up on them and see them. And really, like, some of them weren't that afraid of me. I would just paddle right up to them and they would stare me in the face for 10 seconds. And I would be like, what are you doing? But so I could go out there and I could see them, and I could access the areas, and then of course also look for like behavioral observations that you don't necessarily see when you're electrofishing, especially in reference to like the reproducing aspect that I was talking about. Like I could see the nest, I could see the fry balls, I could see the parents protecting the either the nest or the fry. And over the course of doing this, at least once a week, sometimes multiple times a week, I saw 45 adult snakeheads and hundreds, if not thousands, of eggs and fry. I did not keep track of the eggs and fry just because there were so many of them. But this gives you a little bit of an idea of how it looked. This is really difficult to see. It was much easier in person, but there is a snakehead right here. And so yeah, just same thing, go out, look for them, and just mark the GPS coordinates down when I would find them. This here is a video of what it would look like going out, doing this visual survey. You're gonna see right about here, it looks like the water is almost boiling. And what that is a group of juvenile snakeheads that are still in their fry ball. And so they're kind of like bouncing around the surface, gasping air. And it's quite interesting that, like, to see that. So if you ever see that out there, probably a snakehead fry ball. And of course, later, once I paddled over to this fry ball, I saw both parents hanging out around there. So I do know that they were snakehead fry, and you saw that fish jump over here. Maybe that was caused by the adult snakeheads. I don't really know. But that's just a way that if you see that, you're probably looking at a snakehead fry ball with the boiling water. Now, those were not the only ways that I tried to collect snakehead data. This here is a seine net. And for those of you that aren't familiar with it, basically you have a big pole on this end, a big pole on this end, a long net that stretches in between, floats along the top, and weights along the bottom so that the net stretches out across the whole water column. And so you can just drag this down through the water, loop it around, and then all the fish that are in there are caught, and you can pull it up onto the beach. I tried something similar using seine nets in one of the channels of Otter Point Creek to catch snakeheads. We had one set up down the channel, one up the channel, stretching across the entire creek, walked it down to try to drive the snakeheads into the other net, and then corral them up. We caught just about every fish in that creek except for the snakeheads. We had hundreds of fish of all kinds, but the snakeheads got away from us. We had one jump over the net and get out. There were some other ones that I know were in front of us when we were walking down, but we did not catch. And our theory is that with the dense vegetation on the bottom, the weights maybe just went right over it. So they just went under into the vegetation and just the net went over top of them. But basically, seine nets did not work. And it was pretty labor intensive, and we didn't catch any snakeheads, so I wouldn't really consider that to be a good sampling technique. Another thing that I tried, like I said, I would be in these kayaks and on the stand-up paddleboard, and I would go right up to a snakehead, stare it straight in the face. It would stare back at me. I thought, what if I just had a net and I just scooped it and tried to do that? So it did not work. 2 or 3 times I tried it and realized quickly that this was not going to work. Even if I could get up close to them, as soon as I made a move, they would dart off, and there was no chance to catch them. So really, that was a failed attempt as well. Not going to catch snakeheads that way. Finally, I went out there, and every time I would go out there, I would see people fishing. And I would always say, what are you fishing for? Have you caught anything? They'll say, oh, I'm fishing for snakeheads. Oh, I got one. Oh, I got 2. And I thought, I am no fisherman, but perhaps if I get a fishing rod, maybe I can catch a snakehead or 2. To add to my sample size. I was out there for a few hours on a couple different days, didn't even get a nibble. So for me personally, the fishing rod was a no, but for other people it has worked. So maybe it's something, but certainly for me it was not efficient and did not work at all. So that leaves us with electrofishing, which worked great to get a snakehead in my hands that I could get length, weight, and dissect. And then visual survey worked great to fill in, getting back in those areas where I couldn't get with the boat, and just to kind of fill in the map with where are they located within Otter Point Creek. So both of those were the best methods to sample and collect snakehead data in Otter Point Creek this summer. And that was, remember, the first thing that I wanted to figure out this summer was how best to collect data about snakeheads. So I came to the conclusion electrofishing and the visual survey were the ways that worked for me. The second thing that I wanted to figure out was, what are they eating? So we have some pictures over here. You can see, like, here's a crayfish and here's some 2 little sunfish. And those are some more intact things. Sometimes you just get a scale or sometimes just get a bone. So you can't really tell exactly what it is. Sometimes things are a lot more digested. You can't really tell what it is. But generally speaking, whenever I would get these fish, I would get the stomachs out, cut them open, and see what's inside. So generally, mostly fish is what they were eating. Whether it's eels, killifish, somebody said you just get the bones and the scales, some sunfish, or some fish that are pretty digested and I couldn't tell. That's mostly what we were getting. We did see some crayfish in the stomachs, and really the difference between crayfish and crayfish pieces just was there a head present. If I could find a crayfish head and it was like mostly intact, that's a crayfish. If I just found like a leg or just the tail or like an antenna, then it would be a crayfish piece. And then 2 different times I saw some pieces of plant material in the stomachs. And to be honest with you, I think it's mostly incidental swallowing, because every time we got vegetation, it was a tiny little piece of it, and it was always with something else. It was always with another fish or with crayfish or something else in the stomach. And so I don't really take this as a sign that like these fish are omnivorous or intentionally ingested plant material. I think it's just they just darted for a fish and got a little bit of piece of plant in their mouth as well, and that ended up in their stomach. The second thing, or I'm sorry, the third thing now that I wanted to figure out about these fish is how old they are, and you do that using otoliths. So these otoliths, they're referred to as the ear stones, and they're little mineral deposits that are inside the head of the fish. And so here's a picture of one up close, and here's some different ones of different sizes. These were like tiny ones in a tiny little fish. Most of them were about this size. And what you can do is you can look at rings that are deposited in the otolith and count them like you count tree rings. So what happens is as the fish grows, the otolith gets extra layers deposited on it. But in the winter time, water temperature is colder, everything's just generally slower, the metabolism is slowed down, so not really growing as much, and same thing, the otolith isn't really adding as much either. So you can go and you can see where these like lighter bands are, where the layers have been laid down like really fast, rapidly, and spaced out, versus the winter where they're just like real close, like not much growth. And if you break this in half and polish the one edge, so this would be like this half of it, like in my hand, broken down this way, you can polish it and then look at it under a microscope and see these bands in it. And so these bands are called annulus, plural annuli. And so you see this is 1 winter, 2 winters, looks like there might be a little third line there, 3 winters. And that's basically how you figure out how old this fish is. And so I did that for all the fish that we caught, excuse me, all the fish that we caught. And this is the age distribution that I came up with. So mostly 2-year-olds, fair amount of 3-year-olds, a couple 4-year-olds, 1 5-year-old. But the really interesting thing is that we didn't have any 1-year-olds. And what you would kind of expect is that you would have like this same trend, but continued. So you have a lot of 1, less 2, less 3, less 4, less 5. So this is really interesting to me that in 23 fish, we didn't have a single 1-year-old. Maybe that's just pure dumb luck that we didn't catch any of the 1-year-olds. Maybe something happened to the year class of 2020. I don't really know. So that's something that I thought was really interesting takeaway from this, is that out of 23 fish, no 1-year-olds. Fourth thing, where are they located within Otter Point Creek? So this one here, this map on the right, I'm going to start with this one, are the results from the visual survey. And so each time I would go out in the kayak, stand-up paddleboard, I would keep track of how much time I spent in these areas, like how much time I spent in this channel, how much time I spent in this lagoon, whatever the place may be. And then, of course, I would keep track of, where I saw the snakeheads at, so that I could come back later and say, okay, in this area, I saw this many snakeheads per unit time. So I have it in snakeheads per 30 minutes. And this way, it allows me to kind of standardize the observations that I see in this visual survey, because of the nature of the way the creek is structured, I couldn't spend equal amount of times everywhere. Like to get to these back lagoons, like I have to pass through this. So anytime I come back here, whether I'm going here or whether I'm going here, I'm coming through this every single time there and back. Like I said, the low tide was a bit of a barrier to getting back in here sometimes. So I couldn't spend equal time everywhere, but this allows me to compare, even though I spent, maybe more time over here than I did over here. And what you see is that generally you get a lot more up here in these channels rather than out here in the open. Same thing over here in this map. This map is slightly different because this doesn't take time into account. This is just all the snakeheads that I saw Excuse me. All the snakeheads that I saw all summer long, whether it's boat electrofishing, whether it's I went fishing for them and saw them, whether it was the visual survey, whether they're adults, whether they're fry balls, no matter what it was, they're on this map. And you see a similar thing. They're concentrated up here in these channels by the lagoons, back here in Turtle Creek, not so much out here, not so much out here, all up in here. That being said, there are still some out here. I got one adult here and one fry ball. So they are out here, but just not nearly to the same amount as they are up here in these smaller channels upstream. Fifth thing that I wanted to look at was reproduction. So for the female snakeheads that we call it boat electrofishing, in addition to removing the stomachs and removing the otoliths, I would take their ovaries out. And so here's a picture of some ovaries that I took out of one of the snakeheads, and all these little orange balls are eggs. And so what you can do when you have them is you can take the ovaries out and you can weigh them, and you can calculate something called GSI, which is gonatosomatic index. And that's a ratio of the weight of the ovaries to the total weight of the fish. And what that tells you is essentially, like if there's a higher GSI, it means that for that size of the fish, relative to the size of the fish, there are bigger ovaries, more eggs. Smaller GSI, relative to the size of the fish, again, smaller ovaries, less eggs. And the trend that we see over the course of the summer with the 4 electrofishing trips, trip 1, trip 2, trip 3, trip 4, is that the GSI decreased over the course of the summer. And this makes sense because at the beginning of the summer, you have females that maybe haven't reproduced yet. They're full of eggs. Spawning season's kind of just getting started, versus you get here towards the end of the summer and you see that maybe they've already spawned once, if not multiple times, because that is possible. And so they're kind of out of eggs here, and it's getting towards the end of the spawning season. So we do see that nice trend here. And then this blue line is a linear trend line. Just for statistics sake, 0.74 is the R squared, which is a pretty high R squared value. This is a pretty strong trend that you can see that over the course of the summer, over the course of the spawning season, GSI decreases. As I mentioned, I said I would have some pictures of the nests that I saw out doing the visual survey. I only saw 2 nests over the course of the summer, which is, I guess, kind of a good thing. But I think that just means that I also maybe missed some because they're usually back in these grasses and kind of hard to see. So this is in one of the lagoons, mixed in with some wild rice. And this is a nest here. Here's a drawing to help you orient yourself. So as I said, you get the 2 adults, they come in here, they clip the hydrilla or whatever the vegetation is that floats to the top, and then they lay the eggs in there that float within the clipped vegetation, and then they can hang out in this like open chamber underneath the floating mass of eggs and vegetation. And they're protected from like sight by like these the tall strands that they haven't clipped, and they can kind of hang out down here and keep a watch over the eggs. And so that's what I saw here. You can see some of the clippings, and then all these are eggs all in here. And then underneath here is like an open chamber. And when I did paddle up to this, I did see both adults just take off. So they didn't really do a good job protecting against a large human. They kind of abandoned it at that point, but they were under there protecting the nest, and then they took off when I pulled up, and you could see them like going through the shallow water. When I did find this nest, I would take the eggs out of it if I could. So I just had like a little Ziploc bag, and I would just go and scoop them up to try to get as many of them as I could out of there. And here's what they look like under a microscope. Pretty standard, but just thought it was a cool picture to show. So I only saw the 2 nests, but I did see 12 fry balls of different, ages and life stages. And like I said, that looks like the boiling water from far away, especially when they're older. So these ones that are like small and black, these ones are much younger. These could be a few days old, maybe a week or 2 old. And you just see like a mass of these like little black fish that are all stuck together swimming around. And then sometimes you'll see a parent, maybe both parents, hanging around them, keeping an eye over them. And so I saw lots of them over the course of the summer. These fry over here, are much older. These are probably like a few weeks old. And they start to get this orange gold stripe on the back of them. And they still hang around each other in a general group. They're a little less cohesive than the black ones, the smaller black ones, but they still hang around each other. Parents are still relatively nearby, keeping an eye on them. And so if you ever see them, either these big ones with the orange stripe or the smaller ones in the little black ball, especially if you see the adults nearby, that's a snakehead fry ball. So that wraps up the 5 things that I wanted to look at this summer. And so what conclusions can we draw from all these things that I saw and the data that I collected? First thing, in terms of the sampling method, as I said multiple times, electrofishing was the only way that I caught these fish. Sanding didn't work, the landing net didn't work, the fishing rod didn't work. So the electrofishing was the best way for me personally to get my hands on a fish, to get, length, weight, cut them open, see what's inside, things like that. But that doesn't mean that the visual survey was not very important in providing valuable data, because that allowed me to fill in that map and see where these fish are within Otter Point Creek, which channels they're in. And it also allowed me to see the nests, the eggs, see what the parents are doing, protecting the fry, things like that. For the crayfish, I mentioned that we had the crayfish in the stomachs, and it was still more fish, like it was still mostly fish. But the crayfish that we did have, I got a lot more crayfish in the stomachs than I expected to find. And that was based on a previous study in 2012 that had looked at a lot more snakeheads further down in the lower Chesapeake Bay, but it had found that it was like 97% fish and only 1.4% by number crayfish in the snakehead stomachs. So I was expecting to find mostly fish, which I did, but certainly a lot more fish relative to the number of crayfish that I found. So that's something interesting, is that I'm wondering, is that a continued trend? If I were to keep catching snakeheads, would I continue to see this high proportion of crayfish, or was this something that's a result of only a few, like a smaller sample size? In terms of the age, that was the third thing that we wanted to see. The oldest snakeheads that we had were 5 years old. And so that means that they've probably been here for a few years now. Those 5-year-olds could be the initial ones that swam up the river and established themselves in Otter Point Creek. That's of course possible case, but with the distribution, the number of 3-year-olds, the number of 2-year-olds, snakeheads have been here and reproducing for a few years. And then again, I talked quite a bit about how it's really odd that we didn't get a single 1-year-old fish out of 23 otoliths that I looked at. For distribution, you saw in both of those maps, whether it was the heat map of everything that I saw or the time standardized visual survey one, they're much more frequently found in those smaller upstream channels near the lagoons back in Turtle Creek rather than out in the main area, but they can be out in the main area, just not nearly as often. And finally, in terms of reproduction, I did find evidence of the reproducing population, whether it's the decline in GSI over the course of the summer, showing that the eggs have left the females and they've probably spawned at some point, or whether it's actually like I saw some nests, I saw some fry, so I do know that they're reproducing out there. And this leads us to some directions for future work. So what could come after this study? Something that we had talked about, but I never really was able to get because of time requirements and things like that, would be a fishing tournament. Like I said, I myself am no good fisherman, but perhaps these other people that are out there every week and do catch these fish, maybe they could go out there and add to the sample size as like an organized fishing tournament. Maybe that's something that could work as a sampling method, even though I personally didn't succeed with a fishing rod. A second thing that could possibly come after this is that, like I said, I saw the nests and I saw the fry balls, and whenever I would see them, I would do my best to remove them as much as I could. But there are hundreds out there of fry and eggs, and of course not all of them reach adulthood, but there are lots of nests and fry balls out there. So in terms of limiting snakehead population growth within Otter Point Creek, you could perhaps have some targeted removal of the nests and the fry balls, going out there and scooping them up when you find them. And really, one thing that would just is really important to like solidify some information that I found or see if trends continue is to get a larger sample size. I said I had 23 caught fish, but really that's not that many. That's not as many as I wanted going into the summer. And so when we look at that and we see like, oh, there aren't any 1-year-old fish, if I were to have 100 otoliths to look at, would we still see that? So that's something that's interesting to look at. Like, it'd be really nice if we could continue to get some more fish. And, you know, next summer, if there aren't as many 2-year-olds, then you can say, okay, something happened to the 2020 year class, whether there weren't that many reproduced or a lot of them got eaten. Or perhaps we have 100 otoliths, and when you have a bigger sample size, it evens out and we end up seeing what you expect. But it was just pure dumb luck that I didn't catch any 1-year-olds. I don't know. And that's something that I think would be really interesting to find out. Same thing with the crayfish in the stomachs. I had 23 stomachs. Only 16 actually had food in them. So it's even smaller sample size of stomachs. So when you only have 16 stomachs, it's hard to say like, oh, the snakeheads in Otter Point Creek, they eat more crayfish than elsewhere. And so I really would like to have some more stomachs to get some more stomach contents to see if we had a lot more fish, would it even out where it's like, okay, they do almost entirely eat fish, and it was just chance that I was getting all the ones eating crayfish, or do they really eat more crayfish in Otter Point Creek than elsewhere? It's a great question, and something that we really need a larger sample size for. Finally, I did not do this by myself. I could not have possibly done this by myself. And I owe massive thank yous to a lot of people. So whether it's for funding resources or advice about how to do things, or material resources, space to work, everybody was super helpful this summer and gave me everything that I needed to try to find this information out. So specifically, I'd like to thank Becky, Chris, and Kyle from Steebner for all their help really planning this and getting out in the field with me a few times. Everyone at the Estuary Center, specifically, you know, Christy, Dawn, Lauren, but everybody in general, great. And I can't thank you enough. Kathy and the Otter Point Creek Alliance, Josh Newhart from the U.S. Fish and Wildlife Service, and Catherine Lim from the Virginia Department of Wildlife Resources. Everybody, thank you so much for all your help. And really great time, and I think we found some really cool things. If you're at all interested in some of the papers that I pulled some of that introductory information for, or other information on snakeheads in the Chesapeake Bay in general, here are some of those papers that I cited. And with that, I am open for questions.
All right. So we had one question in the chat, so I could start with that one. And then if anybody else has any, just raise your hand if you'd like to ask Tim directly. So let me roll up and find it. So this one was from Al Marta, and he said that he spends a lot of time in Gray's Run and Cranberry Run. So are you familiar with those areas, Tim?
Unfortunately, no.
They're the other side. If you look it up from a map, they're not out towards the Bush River, but they're up more north.
Gotcha. Coming up the Bush River and taking a right instead of left. Got it. Okay.
And he said he's seen fry balls and he's fished unsuccessfully for snakehead there. The questions are, do they have gills at all or purely air breathers? That's the first.
They do have gills. Yep. And they do breathe oxygen from the water, but they it is part of their biology is that they do have to surface for air, but they can still get oxygen from the water.
Okay. What game fish species do they displace? This is like a three-piece. So what game fish do they displace? Do they really compete with bass, or do they fill a slightly different niche? So
got it. Yeah. So in terms of Otter Point Creek, I can't really answer that specifically because I don't know data on bass populations or anything like that. I do know there was a study, I think it was Potomac. If it wasn't Potomac, it was one of the rivers nearby that looked at snakeheads versus largemouth bass and did try to compare the fish that each of the species would eat. And what it found is that they did have a little bit of overlap, but in that place, they did occupy a slightly different niche in that the snakeheads were mostly eating fish and the bass were, I think, a little bit more generalist. And they were, the different fish species they ate were slightly different. So in that sense, they weren't directly competing with the bass. But I will say there was another study from Blackwater National Wildlife Refuge on the Eastern Shore. And that one didn't really compare snakehead impact in terms of them outcompeting other game fish, but it did look at changes in the fish community pre and post snakehead invasion. So they had some data on the fish community from like 2006, 2007, and then snakeheads showed up in 2012 there, I think. And then they had some surveys from afterwards. And they did find that generally biomass of the fish communities had decreased. There were significant changes in the species there. The dominant species changed. So really, like, I mean, I'm sure you're aware, like everything's interconnected. So maybe they're not necessarily directly competing with the bass, but they, knock out something else, which down the line changes something else, which changes something else. And we don't really know. That's part of this whole thing, is they haven't been here that long. So that's what we need to figure out is, are they going to impact anything? How much, how little, we don't know. But certainly just whenever you introduce a large predator, something is going to get eaten and something's probably going to change.
We need more data. And he said the last one part of his is there any chance of eradication, or are we stuck with them forever?
Unfortunately, I think we are stuck with them forever. Just the number of ones that I saw out there in terms of fry balls, in terms of nests, and just, and unless there was a super, super intensive catch for them, I think we're probably going to be stuck with them, because they're pretty adaptive. They can live in areas that maybe other fish wouldn't survive because of their ability to breathe air. And so I think they're probably going to be here to stay. It's just a matter of figuring out what impact they have. And if we do need to limit their change and distribution, how do we control that?
He mentioned something about a spear, like people using spear guns to kill them,
Mhm.
which I do know down at Patuxent River Park they've done before. Is that something that you guys talked about at all, using spear guns? Or
We had talked about it a little bit. Ultimately, we didn't end up doing that, but I do know there are a lot of people that go out at night. And yeah, they like bow hunt for them or use spear guns. And I think they have quite a bit of success doing that, but that was something that I did not personally attempt to do.
have you ever had, there's one, it's just from a Galaxy Notes, I'm not sure who this is, but it says, have you ever tried a dip net like the one to use for herring? So I'm assuming you used a dip net when you tried to catch it that few times in the kayaks.
Yeah, yes, like I used like it's a little net like that and just tried to scoop, and it didn't work.
Let me see, still scrolling through here. I know I see one hand up so far, so let me just get through the chat. Can they tolerate salinity?
Not a lot. I don't remember exactly what the range is. They're mostly freshwater fish for the most part. They can tolerate a little bit, enough to, move from one river in the bay to another and around. But they certainly aren't something that you would see like out in the middle of the Chesapeake Bay or down, towards the Atlantic Ocean. They're mostly a freshwater fish.
Okay. And if they're showing up on opposite sides of the bay, does that mean they're being introduced on both sides, or are they traveling there themselves?
That's a great question. And I'm not sure if that's known. Like I said, the genetic evidence suggests that they have likely been introduced multiple times. But yeah, to be quite honest, I don't know the answer to that. That's a really good question. I know they can tolerate it for a little bit, so I'm not sure if they can just zoom across the bay and make it over there, or if they were introduced, but yeah, I don't really know about that one.
Yeah, it's interesting. And then the last one, it looks like, well, Kathy Baker-Brosh said we need to have, Otter Point Creek Alliance should host a Snakehead Derby. So
that would be awesome.
All the OPCA people on here. And then Nar and Time, if Southeast Asia doesn't have winter, then how come snakeheads can survive in the American winter?
Right. Well, I guess I should specify, when they're from Eastern Asia, that includes like China, Korea, and like Russia. So they are in areas that do get a little bit cold. They're not necessarily like down in like a tropical, like Vietnam or anything like that. So I think that might answer your question a little bit, just me being a little more specific in terms of their native range.
Oh, and it looks like there was a snakehead tournament scheduled this past summer, but it got moved to Gunpowder River because APG closed the river that day.
Yeah, I do know there was a Gunpowder River one.
Okay, well, we tried, I guess. Try again. Okay, so I think that's everything in the chat. If I missed one, guys, go ahead and post it again. But I see that Doris and Anders has their hand up, so if you want to unmute, you can ask Tim a question.
Hi, this is Sandy Anders, and I'm from Army Environmental Command, Fort Sam Houston, San Antonio, Texas.
Awesome.
And you mentioned, excellent presentation. I loved it. But you mentioned that they fish for them. Do they actually eat them, or are they just trying to get rid of them? Over.
Yeah, people do eat them. I've eaten them quite a few times. So like, whenever all these ones that I would catch electrofishing, of course, I would get like the lengths, the weight, and do all the science stuff to them. But then I'd be left over with these fillets, and they are quite delicious, I will say myself. So among people at the Estuary Center, some other scientists distribute them out, and I've actually seen them come up on menus a few times in restaurants around here. So there's certainly fish that people will eat, and that I will give my humble opinion and say that they taste pretty good, especially with some Old Bay.
Yeah. I haven't tried them yet, but I need to. All
Very
right,
good.
Catherine Benson, or that's Katie. Hi, Katie.
Hi.
Hey.
Great presentation as well. I just had some questions just about the internship in general. So I'm a biology professor at Hartford Community College, and I'm a new member of the OPCA board, or newish. And I was just wondering, what was your favorite part of your internship experience?
For me personally, it was just getting a lot of hands-on stuff. Because of the pandemic, I had a experience the previous summer that had gone entirely online. And so I didn't really get much fieldwork experience there. And then of course my biology labs were all online. So dissecting a fish on a laptop is not the same as doing it in person. So it was really great to just be able to get out there, actually catch a fish. And instead of looking at a diagram on a screen, be like, oh, okay, here's the swim bladder, here's the stomach, here's whatever it is. And so that was really my favorite part, was just the hands-on experience that you can't get any other way by them doing it.
Cool. And then I had another question, if you don't mind. I was going to just ask, like, what advice would you give for someone who might want to apply for this internship, let's say for next year?
Well, I think it was really great that I was able to look on their website, and there were a few like different suggested topics of things that you could look into. But before I had applied, like I talked with Christy and Kyle and Becky and Chris, and so it was really great that like we could like develop and come up with a question that we thought was something that was realistic to look into. So what I would suggest is to like look at the general topics that the Estuary Center is interested in like pursuing and figuring out. But then of course, just look into the literature, figure out some questions that you have that you want to answer, maybe talk it over with the scientists here and see what you can come up with.
Thank you.
Of course.
Any other questions? Because I think we finished the questions in the chat. No more hands are raised. All right. Well, Tim, thank you so much for the presentation today. I know even for myself, I had a lot of things I was curious about. So this was really great to learn more about the snakehead. And yeah, I guess good luck. So can I tell everybody where you're headed?
Sure.
He's going to Denmark for 5 months. So a nice way.
On Friday.
So have fun out there. And yeah, thank you everybody for tuning in.
Yeah,
So,
thank you everybody for showing up. I really appreciate it and listening in.
All right, guys, have a good Sunday. It's almost the end of the weekend, so yeah.